Your mind went quiet an hour ago, but something behind your eyes is still running. The brain doesn't slow down at night. It switches modes. And one of those modes involves flushing the very debris that accumulates when you think, worry, and try to hold things together. Scientists have a name for this system. It only opens fully when you stop being conscious of it. That's the part that changes everything. The nights you feel you're sleeping but wake still fogged. Your cleansing cycle may have run at partial capacity, and you wouldn't have known. Welcome to Sleepy Mind Science. The glymphatic system has been working in the background since the first time you closed your eyes. Tonight, you find out what it's actually been doing. Close your eyes. Feel the weight behind your temples. The process has already begun without you. And now we begin. The brain contains no lymphatic vessels. The system that removes waste from every other organ in the body for decades. Neuroscientists considered this a puzzle with no clean answer. Then, in 2013, researchers at the University of Rochester discovered an entirely separate drainage network woven through the brain, the glymphatic system, named partly after the glial cells that line its channels. This system operates like an internal pressure washer. It uses cerebrospinal fluid, the clear liquid that bathes the brain, to flush waste products out through dedicated pathways along blood vessels. What happens during sleep determines whether it works. The glymphatic system is not always open. During waking hours, glymphatic clearance runs at roughly 5 to 10% of its peak capacity. The brain is too metabolically active, too many signals firing, too much energy being consumed for the channels to operate at full volume. It is only during sleep, specifically during the deeper and later stages, that the system engages fully. Think of it as a building where the cleaning crew can only access the floors once the offices empty out. The brain, in a very real sense, cannot clean itself while you are using it. That operation is reserved for later. Cerebrospinal fluid, the colorless liquid that flows through and around the brain and spinal cord, is produced at a rate of about 500 milliliters per day. Yet the brain holds only about 150 milliliters at any time. That means the entire volume cycles roughly 3 to 4 times daily. During sleep, this cycling accelerates. The fluid moves directionally, flowing inward along arteries and outward along veins, in a pulsing rhythm, driven partly by heartbeat and partly by slow breathing. This pulsation is the glymphatic pump. The calmer and slower your breathing during sleep, the more efficient the pump. The primary target of glymphatic clearance is amyloid beta, a sticky protein fragment that accumulates between neurons as a byproduct of normal neural activity. During waking hours, amyloid beta builds up. During sleep, the glymphatic system sweeps it out and routes it toward the liver for disposal. Studies in both mice and humans have found that even one night of sleep deprivation leads to a measurable increase in amyloid beta concentration in the brain. This is the same protein that aggregates into the plaques found in Alzheimer's disease. Sleep is not passive rest. It is the mechanism that prevents a specific kind of long-term damage. The brain shrinks during sleep, slightly, but measurably. Researchers using 2-photon microscopy in mice found that brain cells, particularly the glial cells surrounding neurons, reduce their volume by up to 60% during deep sleep in animal models. This shrinkage is not damage, it is deliberate. As cells contract, the space between them widens. the interstitial fluid, the fluid that occupies those gaps, can flow more freely. The glymphatic channels effectively become larger, allowing cerebrospinal fluid to move through with less resistance. What feels like rest from the outside is, at the cellular level, an act of opening, making space for the wash that follows. Tau protein, another molecule associated with neurodegenerative disease, is also cleared by the glymphatic system during sleep. Unlike amyloid beta, which forms plaques between neurons, tau forms tangles inside neurons. Both are natural byproducts of neural metabolism. Both accumulate when sleep is disrupted. Research published in Nature Communications found that tau levels in the cerebrospinal fluid rise significantly after a single night without sleep, and that the rise mirrors patterns seen in early Alzheimer's and chronic traumatic encephalopathy. The glymphatic system does not distinguish between these proteins. It sweeps broadly. Sleep determines how thoroughly that sweep runs each night. The term glymphatic was coined by Danish neuroscientist Maiken Nedergaard, whose lab at the University of Rochester produced the landmark 2013 paper that first described the system in living mice. The word combines glial, for the astrocyte cells that line the channels, and lymphatic, referencing the body's peripheral waste clearance network. What Nedergaard's team discovered was that astrocytes, which had long been considered passive support cells, play an active role in pumping fluid through the brain. They do this through specialized water channels called aquaporin, 4 proteins embedded in their membranes. Without these proteins, glymphatic clearance drops dramatically. The cells are not bystanders. They are the mechanism. Not all sleep stages trigger glymphatic activity equally. Slow-wave sleep, also called deep sleep or N3, is the stage most strongly associated with glymphatic flushing. During slow-wave sleep, the brain produces synchronized low-frequency electrical oscillations known as delta waves. These slow oscillations coordinate the pulsation of blood vessels and the movement of cerebrospinal fluid in a way that maximizes flow. REM sleep also contributes, but through a different mechanism. The rapid eye movement stage appears to support the routing of cleared proteins into drainage pathways, leading away from the brain entirely. Both stages matter. Together, they run the complete cycle. The position you sleep in may affect glymphatic efficiency. A 2019 study using dynamic contrast-enhanced MRI suggests that people sleeping on their side, the lateral position, show improved glymphatic clearance compared to those sleeping on their back or stomach. The lateral position appears to align the brain's drainage channels more favorably, allowing cerebrospinal fluid to flow along more natural gradients. This may help explain why side sleeping is the most common sleep position across mammals, including rats, dogs, and most primates. The body may have learned, through evolution, which position gives the brain the best conditions for cleaning by morning. Brain fog, the sensation of mental cloudiness, difficulty concentrating, and slow recall, is not a vague or imaginary complaint. It has a measurable correlate. Research in sleep neuroscience now links brain fog directly to incomplete glymphatic clearance. When the system does not run its full cycle, whether from short sleep, fragmented sleep, or alcohol, residual metabolic waste remains between neurons, interfering with synaptic signaling the effect is experienced as fog because it is. Biochemically, a kind of static, accumulated molecules that interrupt the clarity of neural transmission. Are you missing your brain's nightly cleansing cycle? The fog is one of the answers. Alcohol disrupts glymphatic function even in small quantities. According to animal studies, research published in scientific reports found that in rodent models, low doses of alcohol, equivalent to about 2 drinks, reduced glymphatic transport by roughly 40% compared to alcohol-free sleep. Higher doses reduced it by more than half. Alcohol initially causes sedation, which is often mistaken for good sleep. But the sleep it produces suppresses slow-wave and REM stages, precisely the stages that drive glymphatic activity. The brain may remain unconscious for 8 hours and still wake with a higher toxic load than before sleep. The sensation the following morning has a neurological explanation. Sleep apnea, a condition in which breathing repeatedly stops during sleep, severely impairs glymphatic clearance. Each time breathing pauses, oxygen drops, blood carbon dioxide rises, and the fragile rhythm of cerebrospinal fluid pulsation breaks down. Imaging studies in people with untreated moderate to severe sleep apnea show enlarged perivascular spaces, the channels through which glymphatic fluid flows, suggesting a backup of fluid and reduced clearance efficiency. People with sleep apnea show higher amyloid beta accumulation and elevated cognitive impairment scores, even when matched by age and education, to people without the condition. Treating the apnea partially reverses both findings. The airway and the brain are not separate problems. Glymphatic dysfunction is now considered a possible mechanism linking poor sleep to depression. The prefrontal cortex, the region responsible for emotional regulation, decision-making, and perspective, is among the brain areas most metabolically active during waking and most dependent on overnight clearance. When glymphatic flow is insufficient, inflammatory molecules and metabolic waste accumulate near prefrontal synapses. This blunts their sensitivity and slows their response time. The result is not just cognitive, it affects how quickly the brain can shift out of negative emotional states, how flexibly it interprets ambiguous situations, and how effectively it recovers from stress. Emotional resilience, in part, requires a clean brain. The glymphatic system appears to be more active in younger brains. In adolescence, glymphatic clearance operates at higher efficiency. One reason young people often recover cognitive clarity faster after sleep loss than adults. As the brain ages, astrocyte structure changes. Aquaporin, 4 proteins, which facilitate fluid movement, become misaligned from the blood vessel walls where they function most effectively. This misalignment, documented in aging mouse models and supported by post-mortem human tissue studies, is associated with reduced glymphatic efficiency in older adults. The decline is gradual, and research suggests it may begin earlier than most people expect. Exercise appears to improve glymphatic function through multiple pathways. Animal studies have found that regular aerobic exercise increases the volume of cerebrospinal fluid produced, enhances aquaporin, 4-channel expression in astrocytes, and improves the depth and duration of slow-wave sleep. All 3 mechanisms that support glymphatic clearance. Human research has also found associations between regular moderate exercise and improved overnight brain waste clearance. Though direct glymphatic measurement remains methodologically challenging, The effect appears partially independent of sleep quality, suggesting exercise may support the system's capacity directly. The pathways between movement and brain health are not all biochemical. Some of them flow. Melatonin, the hormone that signals nighttime to the brain, appears to have a direct effect on glymphatic activity beyond simply inducing sleep. Research from 2021 found that melatonin interacts with astrocyte receptors to facilitate aquaporin, function and reduce neuroinflammation in the perivascular spaces. This means that light exposure at night, which suppresses melatonin, does not only delay sleep onset, it may also blunt the glymphatic system's efficiency during the sleep that follows. The body's response to light is not limited to the circadian clock. It reaches into the channels that determine whether the night's cleaning runs completely. The brain produces more than 100 different metabolic byproducts during normal waking function. Glutamate, the brain's main excitatory neurotransmitter, degrades into compounds, including succinate and alpha-ketoglutarate. Serotonin metabolism yields 5-hydroxyindo-acetic acid. Dopamine metabolism leaves behind homovanillic acid. All of these accumulate in the interstitial fluid across the day. The glymphatic system does not target them specifically. It moves the fluid that carries them. But the concentration of these compounds rises with cognitive load, emotional stress, and sleep deprivation. A brain that worked hard today has more to clear tonight. The relationship between effort and rest is chemical before it is anything else. Neuroinflammation, chronic, low-grade inflammation in brain tissue, is both a cause and a consequence of impaired glymphatic function. When the system runs incompletely, inflammatory molecules like interleukin-6 and tumor necrosis factor alpha linger in the interstitial space, activating microglial cells that trigger further inflammation. This creates a feedback loop. Poor sleep impairs clearance, which elevates neuroinflammation, which disrupts the sleep architecture needed for clearance. The loop is not inevitable, but it is self-reinforcing. Breaking it consistently requires both improving sleep quality and reducing the inflammatory load the brain carries into sleep. Which means what happens during the day matters as much as the night. Stress hormones directly suppress glymphatic activity. Cortisol, released during acute and chronic stress, reduces aquaporin, expression in astrocytes, and increases the stiffness of blood vessel walls, reducing the pulsation that drives cerebrospinal fluid movement. Animal studies show that chronically stressed rodents exhibit up to 30% lower overnight glymphatic clearance compared to controls. In humans, evening cortisol elevation, common in people with anxiety, work stress, or unresolved emotional activation, correlates with shallower slow-wave sleep and elevated morning amyloid beta levels. The brain you bring to sleep carries the chemistry of the day you had. That chemistry affects how well the night does its work. The heart's rhythm coordinates with glymphatic pulsation in a way that surprised early researchers. The movement of cerebrospinal fluid through perivascular channels is partly driven by arterial pulse waves, the small pressure changes that propagate through blood vessels with each heartbeat. Slower, larger pulse waves, the kind associated with parasympathetic nervous system activation, deep breathing, and resting heart rate, produce more effective glymphatic flow than the shallow, rapid pulses of an elevated stress response, This is one physiological reason why slow breathing before sleep improves rest. It doesn't just calm the mind. It changes the pressure gradient that drives brain cleaning. Intermittent fasting and caloric restriction appear to enhance glymphatic clearance. Based on research from multiple rodent studies and preliminary human data, eating late at night elevates insulin and disrupts slow-wave sleep, both of which reduce glymphatic efficiency. Conversely, periods of fasting, particularly a 12 to 16-hour gap between dinner and breakfast, allow blood glucose and insulin to fall to levels that support deeper, slow-wave sleep onset. Autophagy, the cellular self-cleaning process that also increases during fasting, appears to work synergistically with glymphatic clearance to reduce the overall metabolic burden on neural tissue. The timing of eating reshapes the chemistry of sleep. Temperature affects glymphatic flow. The brain is one of the warmest structures in the body, and its temperature drops by approximately 1 to 2 degrees Fahrenheit during slow-wave sleep, a drop that appears to facilitate glymphatic activity. Research in animal models published in Science found that brain cooling is not passive. It is actively driven by changes in cerebral blood flow during slow-wave sleep, and that warming the brain experimentally reduces both delta wave production and glymphatic throughput. Sleeping in a room that is too warm can blunt this cooling effect and compress the most glymphatic active sleep stages. The ideal room temperature for brain cleaning runs between 65 and 68 degrees Fahrenheit. Children spend a higher proportion of their sleep in slow-wave stages than adults, which may be one reason why learning consolidation and memory formation are exceptionally efficient in early life. But the glymphatic benefits of slow-wave sleep in children are not limited to learning. The developing brain produces proportionally more metabolic waste per unit of tissue than the adult brain, because neural circuit formation is energy-intensive, and the high ratio of slow-wave sleep compensates. Sleep problems in children, including frequent night waking, delayed sleep onset, and sleep-disordered breathing, carry a specific neurological cost. The clearing cycle that supports development runs shortened. One of the more counterintuitive findings in glymphatic research is the role of norepinephrine during waking hours. Norepinephrine, released from the locus coeruleus deep in the brainstem, suppresses glymphatic activity. This makes evolutionary sense during alert waking. The brain needs to concentrate on external input rather than running internal maintenance. But in people with anxiety disorders, baseline norepinephrine levels remain elevated even during sleep, particularly in the early sleep cycles. This partial waking chemistry reduces the depth of the slow-wave stages where glymphatic clearance peaks. The body tries to rest but keeps one system running that prevents the cleaning from finishing. Across the first 24 facts, a single pattern has emerged. The glymphatic system does not run automatically. It requires conditions: deep sleep, stable breathing, low stress chemistry, side-lying position, a cooled brain, adequate melatonin, and the slow cardiac rhythm of genuine rest. Together, these form the environment in which the night's cleaning can actually complete. What looked like a question about washing the brain turns out to be a question about everything that either enables or interrupts that washing. The facts ahead follow a more specific question, not just whether the system runs, but what determines how completely it runs, and what restores it when it hasn't. Feel the weight of your body where it meets the surface beneath you. Your brain is already in process. The channels are opening, pathways smaller than anything visible, carrying fluid in the rhythm of your own heartbeat. This is not something you can help or hurry. The system knows its work. Let the breath slow on its own. Notice the space between one exhale and the next. That pause is part of the rhythm the cleaning cycle runs on. You don't have to understand it. You only have to remain still long enough for it to continue. And it will. One breath, then the next. REM sleep, rapid eye movement, was first described in 1953 by graduate student Eugene Aserinsky, who noticed that sleepers' eyes moved beneath closed lids in coordinated bursts. His advisor, Nathaniel Kleitman, initially doubted the observation was meaningful. It was. REM sleep turned out to be the stage most associated with dreaming, emotional memory consolidation, and, as later research established, a distinct phase of glymphatic activity focused on routing cleared proteins toward exit pathways. The eyes move because the visual cortex is highly active. The brain is not asleep in any passive sense. It is running a parallel process. During REM sleep, the brain's electrical activity looks almost identical to waking. An electroencephalogram recorded during REM shows high frequency, low amplitude waves, the same signature as alert, focused cognition, yet the person is unconscious and the major postural muscles are paralyzed. This paradox, an active brain in a motionless, unconscious body, led early researchers to call REM sleep paradoxical sleep, a term still used in French and some European scientific literature. The paralysis is deliberate motor cortex activity during REM dreams would otherwise translate into physical movement. The body is held still so the mind can run freely. The first REM period of the night is short, typically 10 to 20 minutes, occurring about 90 minutes after sleep onset. With each successive sleep cycle, REM periods lengthen. By the final cycle before waking, REM can last 45 to 60 minutes. This front-to-back structure means that early sleep deprivation, cutting sleep short by 1 or 2 hours in the morning, disproportionately removes REM sleep. Because glymphatic routing activity is concentrated in REM, and because emotional memory processing depends heavily on REM, the cost of those final hours is not proportional to their share of total sleep time. They carry more weight than they appear to. During REM sleep, norepinephrine, the arousal neurotransmitter that partially suppresses glymphatic function during waking, drops to its lowest levels of the entire 2-4-hour cycle. This near-complete withdrawal of norepinephrine is unique to REM. Researchers at the Nedegard lab discovered in 2019 that this norepinephrine suppression is precisely what allows the glymphatic routing phase of REM to function without norepinephrine holding vessels in a state of slight constriction. Blood vessel walls relax, pulsation amplitude increases, and cerebrospinal fluid can move more freely into and out of deeper tissue. REM cleans differently than slow-wave sleep, but it may clean more completely. The hippocampus, the brain's primary structure for forming new memories, is intensely active during REM sleep. During the day, the hippocampus records events as temporary files, encoding them quickly but incompletely. During REM, it replays these recordings at accelerated speed. Researchers have observed hippocampal time-compressed replay at roughly 6 to 8 times normal pace, transferring encoded experiences to the cortex for long-term storage. This replay is not random. Emotionally significant events are replayed more frequently. The hippocampus appears to be performing a kind of triage, deciding what goes to permanent storage and what fades. The amygdala, the brain's threat detection and emotional processing center, shows its highest activity during REM sleep of any sleep stage, exceeding even waking levels. This elevated activity is not distress, it is processing. Research suggests that REM sleep allows the brain to revisit emotionally charged memories in a neurochemical environment stripped of stress hormones, particularly cortisol and norepinephrine. This creates a condition that functions like emotional recalibration. The charge of a difficult memory is processed and reduced without the chemistry that would re-traumatize. REM sleep is where the brain learns to carry difficult things more lightly. Dreams. The narrative experiences generated during REM appear to serve a memory integration function rather than being the random noise once assumed. Research using targeted memory reactivation found that presenting odors associated with material learned before sleep during REM increased retention of that material by up to 40% compared to presentation during NREM sleep. The sleeping brain, it appears, is actively responsive to cues that direct its consolidation work. Dreams may represent the subjective experience of this sorting process, the brain making associations, testing connections, and discarding what doesn't fit, with the person present as audience rather than director. Lucid dreaming. The state in which a sleeper becomes aware they are dreaming while remaining in REM sleep is associated with unusually high activity in the prefrontal cortex, the region responsible for self-awareness and metacognition. In most REM sleep, the prefrontal cortex is relatively quiet, a feature that may explain why dreams feel real and uncritical. In lucid dreamers, prefrontal activity increases in response to specific eye movement signals, producing a hybrid state. REM's emotional processing and glymphatic routing continue in the background, while a layer of conscious awareness operates simultaneously. The brain can run both systems. Most of us simply don't notice. The default mode network, a set of brain regions that activate during mind wandering, self-referential thought, and daydreaming, remains active during certain phases of REM sleep. This may be why dreams often feel personal, autobiographical, and emotionally weighted. The network that constructs your sense of self continues operating while the regions that evaluate reality are suppressed. Research in cognitive neuroscience suggests that this combination, self-referential processing without reality checking, is precisely what allows dreams to make novel associations that waking cognition, constrained by logic, would not permit. Some creative breakthroughs attributed to dreams may have a neurological explanation. Sleep spindles. Brief bursts of synchronized neural oscillations lasting half a second to 2 seconds, occurring during NREM, stage 2 sleep, act as gatekeepers between sleep stages. They suppress external sensory signals, preventing environmental noise from triggering waking. The density of sleep spindles in a given night correlates with both the depth of subsequent slow-wave sleep and the efficiency of memory consolidation. People who produce more spindles during an afternoon nap show better retention of material learned before the nap than those who produce fewer. Spindle density is partly genetic and partly trainable through consistent sleep scheduling. Slow oscillations. Large, synchronized electrical waves that sweep across the cortex during deep REM sleep, coordinate information transfer between the hippocampus and the neocortex. Each oscillation consists of an up state, during which neurons across the cortex briefly fire in unison, and a down state, a period of near silence. It is during the up states of slow oscillations that hippocampal replays, the time-compressed memory recordings from the day, are transmitted to cortical storage. The architecture of a night's memory consolidation is built out of these paired pulses replay from below, silence above, then cortical integration, the cerebellum, long associated primarily with motor coordination, has recently been found to participate in emotional processing during sleep. Neuroimaging studies show that the cerebellum activates during REM sleep in patterns that correlate with emotional dream content. This finding, reported in Nature Neuroscience, suggests that the cerebellum contributes to the overnight recalibration of emotional responses, not just the fine motor learning it was known for. The brain systems involved in sleep-based repair are more distributed than earlier models assumed. The structure that learned to balance your body is also involved in balancing your emotional memory. The prefrontal cortex undergoes a specific kind of maintenance during slow-wave sleep. During waking, prolonged cognitive effort causes a gradual buildup of adenosine, a metabolic byproduct of neural activity that acts as a natural sleep pressure signal. Adenosine accumulates fastest in the most active brain regions. In people who work under sustained cognitive load, prefrontal adenosine levels are elevated, and the sensation is experienced as mental fatigue, difficulty focusing, slower decision-making. To reduced impulse control, slow-wave sleep clears adenosine preferentially from high-load regions. The prefrontal cortex is often the first region to recover after good sleep and the first to degrade without it. Chronic sleep restriction, sleeping 6 hours per night for 2 weeks, produces cognitive impairment equivalent to 2 full nights without sleep. But with a crucial difference, people who are chronically sleep restricted cease to notice their own impairment. They adapt to a lower baseline and evaluate themselves as functioning normally. This subjective normalization has been documented in controlled studies, where participants reported feeling fine while performing at the level of severely sleep-deprived controls. The brain's self-monitoring systems, located in the prefrontal cortex, are precisely the systems most impaired by sleep loss. So the instrument used to assess the damage is itself damaged. The relationship between sleep and insulin sensitivity reveals another layer of how overnight brain repair connects to metabolic health. Slow-wave sleep is the stage during which growth hormone, which drives tissue repair throughout the body, is secreted most intensely. Growth hormone also suppresses insulin secretion and promotes fat oxidation. When slow-wave sleep is disrupted, growth hormone release is blunted, insulin sensitivity decreases, and the brain's own glucose metabolism shifts. Given that glucose is the brain's primary fuel, this metabolic disruption compounds cognitive impairment. The brain wakes with less effective fuel delivery as well as a higher residual waste load. Both require the next night to partially correct. You are at the midpoint of the night. The first half of what your brain needed to do, it has already begun. The slow waves have moved through. The fluid has flowed along channels that require nothing from you except this stillness maintained long enough for the cycle to continue. Let the body drop a little further. The spine, the hips, the backs of the knees, There is no effort required here, only the absence of interruption. The second half of the night does its work in the same way the first half did, without being asked, without being noticed, in the space you leave for it. Stay here. That is the whole instruction. The glymphatic system continues to operate during anesthesia, which initially suggested that unconsciousness alone was sufficient to trigger it. Later research clarified the mechanism the specific anesthetic agents used, particularly ketamine and xylazine, happened to mimic the slow oscillatory brain states of deep natural sleep. Other anesthetics, including isoflurane, do not produce the same effect and show reduced glymphatic activity. The finding confirmed that it is not unconsciousness per se that enables glymphatic function. It is the specific electrical pattern of slow-wave oscillation that opens the channels. The brain's cleaning mode has a signature. Blood-brain barrier permeability fluctuates across the sleep-wake cycle. Research published in Nature Neuroscience found that the barrier, which controls what enters the brain from the bloodstream, becomes slightly more permeable during slow-wave sleep in specific regions, allowing larger molecules to enter for nutritional delivery while the glymphatic system simultaneously clears the space for them. This coordinated opening and clearing is time-sensitive. It depends on the oscillation cycle being maintained. Fragmented sleep disrupts the timing, reducing both delivery and clearance without necessarily shortening total sleep duration. Traumatic brain injury disrupts glymphatic function in ways that persist long after the acute injury resolves. Research in animal models shows that even mild concussion reduces aquaporin 4 expression and alters perivascular space geometry for weeks to months after impact. This may help explain the prolonged cognitive symptoms: brain fog, fatigue, emotional dysregulation that follow concussion in humans. The glymphatic system needed for recovery may be impaired by the very injury it needs to help heal. Sleep is prescribed for concussion recovery not only for rest, but because it is the primary window for glymphatic-driven repair. Intermittent hypoxia, brief drops in blood oxygen during sleep-disordered breathing, damages the perivascular channels through which glymphatic fluid moves. Even mild, repeated oxygen drops produce oxidative stress in the astrocyte end feet that line these channels, reducing their flexibility and fluid channeling efficiency. This damage accumulates with each disrupted night. Unlike the reversible suppression caused by stress hormones or alcohol, the structural changes from chronic hypoxic injury respond more slowly to intervention, sometimes taking months of treated sleep before glymphatic efficiency measurably recovers. The timeline of repair follows the timeline of damage. Light plays a regulatory role in glymphatic function beyond simply triggering melatonin release. Intrinsically photosensitive retinal ganglion cells, a class of retinal cells containing melanopsin, are most sensitive to short-wavelength blue light in the 480 nanometer range. These cells project directly to the suprachiasmatic nucleus and also to the paraventricular nucleus, which regulates autonomic nervous system tone and indirectly affects glymphatic pulsation via cardiac rhythm. Blocking blue light in the 2 hours before sleep reduces sympathetic activation, slows heart rate, and shifts the pre-sleep chemistry toward conditions that support glymphatic flow. The eye is not just for seeing. Omega-3 fatty acids, particularly docosahexaenoic acid, directly support glymphatic function. DHA is the primary structural fatty acid in brain cell membranes. including the astrocyte end feet that line glymphatic channels. Studies in aged rodents have found that DHA supplementation restores aquaporin 4 alignment toward baseline young adult levels and reduces overnight amyloid beta accumulation. Dietary DHA primarily comes from fatty fish, algae, and some fortified foods. Its relationship to sleep is not through sedation. It acts structurally, maintaining the integrity of the channels that determine whether the night's cleaning runs efficiently. The vagus nerve, the long wandering nerve that connects the brainstem to the heart, lungs, and gut, carries information about the body's physiological state back to the brain. Vagal tone, a measure of parasympathetic nervous system dominance, correlates positively with slow-wave sleep depth and glymphatic activity. High vagal tone is associated with slower heart rate, greater heart rate variability, and more efficient overnight brain clearance. Practices that improve vagal tone: slow exhalation, humming, cold water exposure to the face, and consistent sleep scheduling measurably affect the quality of the brain's overnight maintenance cycle. The relationship between gut microbiome composition and sleep quality is now supported by multiple bidirectional studies. The gut produces approximately 90% of the body's serotonin, a neurotransmitter that influences mood, appetite, and, through its conversion to melatonin in the pineal gland, sleep timing. Disruption of microbiome diversity from antibiotics, poor diet, or chronic stress reduces serotonin production, blunts melatonin output, and shifts sleep architecture toward lighter stages. Conversely, sleep deprivation alters microbiome composition within days, reducing populations of lactobacillus and bifidobacterium strains associated with GABA production and inflammatory regulation. Chronic pain impairs sleep architecture through multiple mechanisms. Pain activates the locus coeruleus, raising norepinephrine and suppressing glymphatic activity. It produces frequent microarousals that prevent slow-wave sleep from reaching the depth needed for full delta wave production. And it creates a sensitization loop. Poor sleep lowers pain thresholds the following day, which worsens pain, which further disrupts sleep. Research in fibromyalgia and lower back pain patients shows that treating sleep through cognitive behavioral therapy for insomnia reduces pain intensity ratings independently of any analgesic intervention. Sleep quality is a pain management variable. The thalamus acts as a gatekeeper between the senses and the sleeping cortex. During NREM sleep, thalamic neurons switch from transmission mode to oscillation mode, generating the sleep spindles that block external sensory signals from reaching cortical awareness. This sensory gating is what allows you to sleep through moderate ambient noise. It is also fragile. Thalamic gating efficiency degrades with age, explaining why older adults wake more easily. The same system is disrupted by anxiety, where thalamic spindle production is reduced. Environmental sounds penetrate more easily, and sleep remains shallow. The gatekeeper gets tired. Ketone bodies, produced during periods of low carbohydrate intake or fasting, appear to support slow-wave sleep architecture. Research in metabolic science found that mildly elevated ketone levels, such as those reached after a 12 to 16-hour fast, correlate with deeper slow-wave sleep and higher delta power on electroencephalograms. The mechanism appears to involve ketones serving as a preferred fuel source for astrocytes during the metabolically intensive maintenance work of deep sleep. The link between fasting, ketosis, and sleep quality offers one nutritional pathway by which metabolic choices may directly affect glymphatic efficiency. Yoga Nidra, a guided relaxation practice that targets the hypnagogic threshold between waking and sleep, produces measurable changes in brain state associated with improved sleep quality. Research published in the International Journal of Yoga found that regular practitioners of Yoga Nidra show increased dopamine levels, reduced cortisol, and higher theta wave activity during the practice. While not equivalent to slow-wave sleep, the physiological shift toward parasympathetic dominance achieved in Yoga Nidra creates pre-sleep conditions that reduce sleep latency and deepen the first slow-wave cycle, the most glymphatic-rich of the night. The suprachiasmatic nucleus, the brain's central clock, can be partially reset by non-photic cues in addition to light, social cues, meal timing, and exercise timing all influence circadian phase, though less powerfully than light. Research in shift workers found that combining light therapy with timed meals and scheduled exercise could accelerate circadian re-entrainment and improve glymphatic phase-relevant deep sleep timing after shift changes. More effectively than any single intervention alone, the clock responds to a coordinated system of inputs. Adjusting one input without the others produces partial results. The hippocampus has a functional limit on how much new information it can hold before requiring consolidation. Researchers estimate that the hippocampus can maintain approximately 24 hours of declarative memory before its capacity saturates. Sleep is not optional for learning. It is the mechanism by which space is made for the next day's encoding. Studies that deprive subjects of sleep after a learning session show 40 to 50% lower retention after 1 week, compared to those who slept normally following encoding. The classroom and the bedroom are metabolically linked. Seasonal variation in sleep quality is driven partly by changes in light exposure and partly by direct temperature effects on slow-wave sleep induction. In winter, people at higher latitudes tend to sleep longer but report lower sleep quality, a pattern consistent with reduced light exposure, suppressing daytime cortisol peaks and thereby reducing the adenosine gradient that drives sleep pressure at night. Vitamin D deficiency, which becomes more common in winter at northern latitudes, has also been independently linked to reduced slow-wave sleep and elevated nighttime waking, though the mechanisms are still being characterized. The pineal gland was called the seat of the soul by philosopher René Descartes in the 17th century. The one structure he believed was not duplicated in both hemispheres, and therefore the location where the unified mind resided. He was wrong about the soul, but right about the asymmetry. The pineal gland sits at the midline of the brain, unpaired. Its melatonin production, timed by the suprachiasmatic nucleus through a sympathetic neural relay, has since been mapped in precise neurochemical detail. Descartes was looking at the right structure for the wrong reason. The pineal controls the chemistry of night. Cortical thinning, a gradual reduction in the density of the gray matter layer covering the brain, accelerates with chronic sleep loss. Neuroimaging studies tracking adults over 5 years found that those who consistently slept less than 6 hours showed significantly greater cortical thinning in the frontal and temporal lobes compared to adequate sleepers. These regions are responsible for language processing, executive function, and emotional regulation. Cortical thinning is not immediately reversible with recovery sleep. It represents a structural consequence of years of incomplete glymphatic clearance. The bill arriving late. A 2023 study from the Karolinska Institute found that a single night of total sleep deprivation was associated with a 17% increase in tau protein in the cerebrospinal fluid, and that this elevation persisted for 48 hours after recovery sleep. This extended persistence suggests that even short-term acute deprivation leaves a residue that sleep alone cannot fully resolve within a single subsequent night. The brain's debt collection system is slower than its debt accumulation system. One recovery night begins the repair. The completion takes longer. Dreaming in color, as opposed to dreaming in black and white, is more common in people who grew up watching color television and rare in people who grew up with black and white only. Research by psychologist Eva Merzin found that adults over 55 who had access only to black and white television in childhood reported dreaming in monochrome in roughly 25% of their dreams, compared to nearly zero in younger adults. The visual content of REM is partly shaped by the visual diet of waking life. The screen shapes the dream. The sleep of whales and dolphins demonstrates that the glymphatic system's architecture has been solved differently across evolutionary lineages. Cetaceans practice unihemispheric sleep. One cerebral hemisphere sleeps while the other remains conscious, allowing continued surface breathing and partial alertness. Electroencephalogram recordings from bottlenose dolphins show slow waves in one hemisphere, while the other displays waking-like patterns simultaneously. Whether each hemisphere runs its own glymphatic cycle independently during unihemispheric sleep is still under investigation. But the dolphin's brain does appear capable of maintaining alertness and conducting maintenance at the same time. Hypnic jerks. The sudden muscle contractions that jolt the body at the edge of sleep onset are not signs of a problem. They occur during the transition from wakefulness to NREM stage 1, when motor cortex activity briefly fires as inhibition descends. One theory holds that they evolved as a startle prevention mechanism, the brain testing motor readiness before fully surrendering voluntary control. They are more common when sleep onset occurs quickly, under fatigue, or after caffeine. Most people experience them several times per week without awareness. The phenomenon of sleep paralysis. The state of wakefulness accompanied by inability to move, occurring at REM onset or offset, affects an estimated 8% of the population at least once. It occurs when the REM atonia system, the motor suppression active during dreaming, fails to switch off simultaneously with the return of consciousness. It is often accompanied by vivid hypnagogic hallucinations and a sense of presence in the room. Historically, sleep paralysis has been reported across cultures as supernatural visitation, the old hag in Newfoundland, the incubus in medieval Europe, the kanashibari in Japan. Shift work disorder. A circadian rhythm disorder affecting people who work outside traditional daytime hours, is classified as a medical condition by the International Classification of Sleep Disorders. It affects an estimated 26% of shift workers and is associated with elevated cancer risk, metabolic syndrome, cardiovascular disease, and accelerated cognitive decline. The mechanisms involve chronic glymphatic disruption, prolonged elevated cortisol, reduced slow-wave sleep time, and persistent circadian misalignment. Medical recognition of the disorder has prompted some health systems to classify it as an occupational health risk, equivalent to other chronic workplace exposures. Sleep spindle density. The number of sleep spindles produced per hour of REM stage 2 is one of the strongest correlates of general intelligence identified by sleep science. Studies in both adolescents and adults find that higher spindle density is associated with better performance on fluid intelligence tasks, working memory tests, and academic outcomes. Because spindle density is partly heritable, but also responsive to sleep regularity and stress reduction. It represents one of the few measurable sleep variables that is both an outcome and a potential target for cognitive maintenance. The brain that cleans well, remembers well, feels clearly, and recovers quickly is not a different brain from the one that doesn't. It is the same brain, given conditions the glymphatic system requires to complete its work. What these 75 facts have established is not a list of sleep advice. It is a description of a mechanism specific, biological, and conditional. The remaining facts follow a different register. They do not add more mechanism. They ask what it means to know this, and what it would take to actually let the night do what it is designed to do. You have passed the middle of the night. Whatever the day left in you, the decisions unmade, the conversations still running. None of it requires anything of you right now. The channels that carry the debris of waking life are open. The fluid that moves through them is moving in the rhythm of your own slowed heart. Let the jaw go. Let the hands open. Let the breath find its own pace without you steering it. The glymphatic system does not need your attention to run. It needed your stillness. You have already given it that. What it does next, it does without asking. The night already knows what to do. The brain during deep sleep is quieter than at any other moment. Not silent, but quiet in the way that a room is quiet when the work is finally being done rather than discussed. Delta waves move through at 1 cycle per second. Each cycle opens the space between neurons a little wider. The fluid flows. Something is being carried away that accumulated over the course of a life that kept trying. Adenosine, the sleepiness signal, is made from ATP, the molecule every cell uses for energy. The more a neuron fires, the more ATP it burns. The more ATP it burns, the more adenosine it leaves behind. Waking is metabolically expensive. Sleep is the interval in which the cost is paid. Not metaphorically, biochemically. The brain spends all day accumulating what the night is built to clear. REM sleep was running in you long before you were born. Fetal brain recordings show REM-like activity from as early as 23 weeks of gestation. In the 3rd trimester, fetuses spend up to 80% of their time in REM-equivalent states. The developing brain appears to use this stage to wire itself, to test neural connections before they are needed. You began processing experience before you had any. The night has been working for you longer than memory can reach. The slowest heart rate of your life occurred during one of your deepest sleep cycles. Parasympathetic tone peaks in slow-wave sleep, dropping heart rate and blood pressure to their lowest points of the 2-4-hour period. This is the body's most protected state, the window in which cardiovascular recovery occurs. Arterial walls relax, and the rhythm that the day disrupted resets itself quietly. The heart rests most completely in the dark. There is a specific kind of forgetting that only happens during sleep. The hippocampus, while consolidating memories, also prunes associations. It removes links between unrelated events that happened to co-occur. Without this pruning, every experience would chain to every other experience, creating a tangled web rather than a usable map. Sleep doesn't only preserve what matters, it selectively removes what doesn't. The clarity of memory depends as much on what the night erased as on what it kept. Slow-wave sleep generates something called sharp-wave ripples, or brief, high-frequency oscillations in the hippocampus that occur during the up states of slow oscillations. These ripples are the signal by which the hippocampus sends memory traces to the cortex for long-term storage. They last less than 100 milliseconds each. In a single night's sleep, thousands of them fire. Each one is a small transfer. Across a lifetime of sleep, they are the architecture of everything you know. The brain's default mode network, active during rest, self-reflection, and mind-wandering, has a cleaning schedule. It is among the regions with highest resting state metabolic activity, meaning it accumulates waste quickly. Glymphatic flow through default mode regions is most efficient during slow-wave sleep, which is also when the network is least active. The system that builds your sense of self quiets so the night can maintain it. Identity, at the cellular level, requires maintenance. Emotional memories are stored differently from factual ones. Factual memory, what you had for breakfast, a name, a phone number, is primarily hippocampal. Emotional memory, the feeling tone attached to an experience, involves the amygdala, which processes and stores effects separately from narrative. REM sleep strengthens emotional memory by stripping the stress charge from the amygdala's encoding. The facts fade. The feelings, processed in the dark, become something the body carries differently by morning. The concept of memory reconsolidation, the finding that every time you retrieve a memory, you briefly destabilize it and restore it, means that sleep following a significant memory retrieval is neurologically important. If you revisit a painful memory before sleep, REM may reduce its emotional charge during consolidation. If you revisit it while sleep-deprived, reconsolidation is incomplete, and the memory restores with its full effective intensity. How you sleep after a difficult day changes what that day becomes. Brainwaves during sleep were first recorded in 1937 by Alfred Lee Loomis, who used an electroencephalograph to classify sleep into distinct stages. He identified the slow, rolling waves of deep sleep, what we now call delta waves, and the faster, smaller waves preceding them. He was measuring what is now understood to be the opening and closing of the glymphatic system, a pattern invisible to him, legible to us. Science sees what its instruments can reach. The word somnambulism, sleepwalking, derives from the Latin for sleep and walk. It occurs during slow-wave sleep, not REM. The motor system partially activates while the cortex remains in deep sleep, producing coordinated movement without conscious awareness. Sleepwalkers cannot form new memories of their actions because hippocampal recording is suppressed. They navigate by habit and procedural memory. The architecture of the sleeping brain is not a single state. It is a layered system that can come partially online without waking. The brain's waste drainage reaches the lymphatic system of the neck, cerebrospinal fluid. After moving through the glymphatic network, exits the brain through meningeal lymphatic vessels. Discovered in 2015 by Jonathan Kipnis at the University of Virginia, and drains into cervical lymph nodes. These vessels deteriorate with age, contributing to the age-related decline in glymphatic clearance. Research is underway to test whether interventions that maintain meningeal lymphatic vessel health, including exercise and anti-inflammatory therapies, can slow this component of the decline. Conscious relaxation. Deliberately releasing muscle tension before sleep improves glymphatic flow by reducing baseline sympathetic activation. Progressive muscle relaxation, body scans, and slow exhalation-focused breathing each reduce heart rate and cortisol, shifting pre-sleep chemistry toward the conditions the glymphatic system requires. The practice doesn't add hours. It improves the quality of the hours already there. 5 minutes of deliberate release before sleep changes the chemistry of what follows. The last hour of sleep is not a bonus. It is a completion. REM cycles lengthen toward morning. The final cycle before waking contains more REM than the first 3 combined. Emotional processing, memory routing, and the specific glymphatic work of REM all complete their largest phase in these final hours. Cutting sleep short by 1 hour removes disproportionately from this phase. The last hour of sleep is not extra. It is often the most restorative of all. Fibromyalgia, a condition involving widespread pain, fatigue, and cognitive disruption, is characterized by an abnormality in slow-wave sleep alpha wave intrusion, where waking frequency alpha waves break into the delta wave pattern of deep sleep, preventing it from reaching full depth. The result is sleep that feels ongoing but provides incomplete glymphatic clearance and inadequate restorative chemistry. The sensation of waking exhausted after a full night of sleep has, in fibromyalgia, a measurable neurological signature. The sleeping brain can still respond to its own name. Auditory processing during sleep is not eliminated. It is filtered. Familiar, emotionally significant sounds. Your name, a baby crying, a fire alarm, pass through thalamic gating more easily than neutral sounds. Research using polysomnography showed that hearing your own name during NREM sleep produces a distinctive K complex, a large, brief wave indicating unconscious processing. The sleeping brain listens. It simply decides what to act on. Growth hormone released during slow-wave sleep affects not only body tissue repair, but also brain plasticity. Research indicates that growth hormone promotes the production of new myelin, the insulating sheath around neurons that determines how quickly they conduct signals. Myelin repair and remyelination are ongoing throughout life, and impaired slow-wave sleep reduces this process. Cognitive decline associated with aging may be partly driven by cumulative myelin maintenance failures that accumulate across decades of shortened deep sleep. The body's core temperature reaches its lowest point around 4 a.m. in people following a conventional sleep schedule, approximately 2 to 3 degrees Fahrenheit below peak afternoon temperature. This nadir coincides with the deepest and most glymphatic active sleep. Waking before this nadir from an alarm, a noise, or anxiety interrupts the deepest maintenance phase of the night. The body's internal thermometer and the brain's cleaning cycle are scheduled to align. They share the same window. The brain of a person who has just fallen in love shows REM sleep disruption, not from happiness, but from the elevated norepinephrine associated with romantic arousal. New love produces a neurochemical state similar to stress in its suppression of glymphatic function. This may be partly why early romantic relationships are associated with sleep difficulties and morning grogginess. Not from lack of sleep, but from its reduced restorative quality. The glymphatic system does not distinguish between pleasant and stressful arousal. Both interfere. Music tempo affects sleep onset and slow-wave sleep architecture. Research found that music at 60 to 80 beats per minute, close to resting heart rate, facilitates sleep onset by entraining cardiovascular rhythm and reducing arousal. Slower tempos continued during early sleep may extend the transition into slow-wave stages. Music above 100 beats per minute maintains sympathetic activation and delays the cardiac deceleration that glymphatic onset requires. The rhythm you bring to the threshold shapes the descent. The olfactory system is the only sensory system with direct access to the hippocampus and amygdala without passing through the thalamus. Smell bypasses the sleep gatekeeper. Research using olfactory stimulation during sleep found that odors associated with positive memories presented during slow-wave sleep. Enhanced consolidation of associated learned material. The finding has practical implications for memory therapy and is consistent with the broader principle. The sleeping brain is not closed. It is selectively open. Magnetic resonance imaging studies of the brain during slow-wave sleep show that the cortex does not go uniformly dark. Local slow waves, delta oscillations confined to specific brain regions, occur independently, based on which areas worked hardest during the preceding day. The brain's maintenance schedule is personalized. A musician practices and their auditory and motor cortex local slow waves deepen. A writer reads and their language network clears more thoroughly. The night repairs specifically what the day built. People with high sleep efficiency, the ratio of time asleep to time in bed, show lower lifetime risk of developing Alzheimer's disease in prospective studies, independent of age, education, and genetic risk factors. The mechanism runs through glymphatic amyloid clearance. Each percentage point of sleep efficiency loss is not dramatic on its own. Accumulated over decades, it determines the clearance margin between a brain that maintains itself and one that gradually does not. The meningeal lymphatic vessels, the channels through which glymphatic drainage exits the brain, can be stimulated by low-level transcranial photobiomodulation light-based therapy applied to the skull. Early clinical research found that near-infrared light penetrating the skull improved lymphatic drainage efficiency and reduced amyloid burden in mild cognitive impairment patients. The technique is years from clinical readiness, but the finding confirms that the brain's drainage pathways are accessible from outside and responsive to intervention. The walls are thinner than they appeared. The brain that cleaned itself well tonight carries less forward than the one that didn't, not because it thought less, or felt less, or tried less, but because it was given the conditions in which the system designed for its maintenance could actually finish its work. The answer was never willpower or sharper attention. It was always the same night's work, done completely. The night does not ask you to improve. It only asks that you stop standing in the way of what it already knows how to do. The channels that open tonight, the cerebrospinal fluid moving through spaces smaller than a strand of hair, the amyloid carried away from the places it accumulates, the norepinephrine finally falling so the routing could begin. None of that required anything of you except being here long enough for the cycles to complete. We followed the glymphatic system from its first description in a Rochester laboratory to the meningeal vessels at the base of the skull. We watched norepinephrine fall so REM could begin its own kind of clearing. What began as a question about fog became a map of the machinery underneath it. The brain was never passive. It was always building toward the moment when you would finally stop. Relaxing facts about sleep has been following the chemistry of the night across many hours. What the brain requires. What disrupts it? What restores it? The next video in this collection follows what happens to that chemistry under the weight of a life that keeps moving too fast. That video is waiting whenever you are ready. Something is already releasing. The channels are open, and the fluid is moving in the rhythm of your own slowed breath. Let the body settle into the surface beneath it. Not the weight of effort, just the weight of being finished for now. If sleep comes, let it take you into the phase where the real work continues without you. The system that repairs you does not need your attention. It only needed you to stop. But if your eyes are already closed, stay there. Rest well. Quiet mind. The fog you carried in has somewhere to go now, where the science of sleep becomes the softest place to land. Good night.